Hello guys, welcome everyone to this evening session on DNA replication। आप लोग का स्वागत है लक्ष्य JRF सीरीज़ में। और इस सीरीज़ में हमारा स्टार्टिंग टॉपिक होगा डीएनए रेप्लिकेशन। मैं सभी लोगों को वेलकम करता हूँ ऑनलाइन, जितने भी स्टूडेंट्स हैं। आई वेलकम एवरीबॉडी ऑनलाइन। सो प्लीज़ गिव अ थम्स अप एंड so that I can know that you are online. And guys, please like, share and subscribe this video as much as possible because आपके efforts बहुत ही important हैं हम लोग के लिए। And please be regular in this class. So this is a starting class. So आज की class starting class है DNA replication की। So I'm beginning the class. So class begin करने से पहले मैं आप लोग को बताना चाहूँगा that please go to the Google Play Store and in the Google Play Store you need to install the Unacademy Learning app and then you need to select your goal. In the select your goal category you need to select CSIR UGC NET. So once you select CSIR UGC NET, so you will be getting access to various top educators courses that is available for free in the Unacademy app. And if you want to go for more, if you want to go for exclusive coverage of the syllabus, if you want to go for if you want to go for exclusive coverage of the syllabus, if you want to go for live interaction with the educator, and if you want to go for well planned classes, then you should go for 12 months to 24 months of subscription. Although the subscription is available for 1 month, 3 months, 6 months, but जो ideal subscription है that is available for 12 months to 24 months because पूरे साल का कोर्स डिज़ाइन होता है एंड आपको यहाँ पर अच्छे से पूरे साल की कवरेज आपको मिलती है। So this class is important because taking 12 months to 24 months of subscription is very important because ये क्लासेस होती हैं पूरे साल भर चलती हैं और अगर आप एक भी क्लासेस को सब्सक्राइब करते हैं तो आपको जितने भी एजुकेटर्स हैं सबकी क्लास का आपको एक्सेस मिलता है। सो दैट इज़ द एडवांटेज। प्लस आपको नोट्स मिलते हैं, प्लस आपको मॉक टेस्ट मिलते हैं, क्विजेस मिलते हैं, बहुत सारी चीज़ें मिलती हैं। सो दिस इज़ एन एडवांटेजेस सिचुएशन फॉर एन एस्पायरेंट। और 12 मंथ्स का टाइम बहुत अच्छा टाइम होता है। इसमें 2 बार आप अच्छे से प्रिपेयर कर सकते हैं किसी भी एग्ज़ाम जो कि 2 बार होता है साल में। So let's begin our session. I hope everybody is there online. Then you need to confirm the payment details and you can enter my code HY10 for getting 10% off. So the entire course will be available for 10,395 instead of 11,550. And you can do the payment by debit card or credit card or by EMI, and these EMI are all no cost EMI. You can also do the payment by net banking. So that is also the topic for today is DNA replication, and I welcome everybody to Lakshya JRF 2021. So this is a new series which has been started. So we are offering courses for the life sciences, chemical sciences, mathematical sciences so far. So Amit sir and Rahul sir will be hosting classes for the mathematical sciences. Chemical sciences will be taken by the Richa ma'am, and life sciences में myself Himanshu Yadav, and I will be coordinating with Kumar ma'am, which is also faculty, who is also faculty for life sciences. So these two people will be telling you about life sciences and will be teaching you about the details of the syllabus. And so let's begin today's class. So the topic is DNA replication. So this is a very basic topic and it is a very important one. So basically it is a process in which a single DNA molecule leads to the formation of two daughter DNA molecule. So this occurs in the S phase of the cell cycle, and it is a very important process. So let's see the structure before beginning in the beginning. So there is deoxyribonucleotides that are the bases which are basically adenine, guanine, cytosine and thymidine, and in this the nitrogenous bases they are attached to the phosphate group, So there is deoxyribose sugar, then there is a nitrogenous base, and then there is a high energy triphosphate group. So these three all together, they are known as nucleotides, and in this case it is deoxynucleotide. But in the other case also there is ribonucleotides also, in which there is ribose sugar instead of deoxyribose sugar. So ribose sugar is present in case of ribonucleotide. So in that case there is uracil which is present instead of thymidine, and high energy triphosphates are present. So this is the structure of deoxyadenosine triophosphate, and this is the structure of this one is the structure of deoxyadenosine triphosphate, which is also known as DATP. So deoxyadenosine triphosphate and deoxyguanosine triphosphate, DATP and DGTP, are the purines. So they are the purines, and DCTP and DTTP are the pyrimidines. So there is a difference between purines and pyrimidines. So cytosine and thymine are the pyrimidines, and adenosine and guanosines are the purines. So this is the 5 prime, and this is the one strand of DNA that is arranged in the 5 prime to 3 prime direction, and there are phosphodiester bonds which are present between the deoxyribose sugar molecule. So these are the phosphodiester bonds. And this is the one strand of the DNA which is present in the 5 prime to 3 prime direction. Now structure, it's like the structure of the DNA is like one strand goes in 5 prime to 3 prime direction, and the other strand moves in the 3 prime to 5 prime direction, so that is in the anti-parallel manner. And thymine and adenine are joined by 2 hydrogen bonds, whereas cytosine and guanine are joined by 3 hydrogen bonds. So these are the hydrogen bonds. These are the hydrogen bond that is taking place between the nitrogenous bases. This one is the hydrogen bond. This one again is the hydrogen bond. So one strand is arranged in the 5 prime to 3 prime direction, and another strand is arranged from 3 prime to 5 prime direction. So it is anti-parallel. And then there are phosphodiester bonds between two. These are the phosphodiester bonds. These are the phosphodiester bonds which are present between two nitrogenous sugar molecules which are attached to the nitrogenous bases. So this is an ideal structure of DNA molecule. Then there is a region of major groove, and then there is a region of minor groove. So this one is the region of major groove. This is the region of the minor groove. And there are 10.5 bases per ton of the helix. So there are 10.5 bases per ton of the helix, and these are the bases which are paired in the middle. Then there is a region of major groove, and then there is a region of minor groove. So this one is the region of major groove. This is the region of the minor groove, and there are 10.5 bases per ton of the helix. So there are 10.5 bases per ton of the helix, and these are the bases which are paired in the middle. Then there is strands of opposite polarity. and then there is right-handed helix orientation and left-handed helix orientation. So there is A form and B form which is in the right-handed helix orientation, and then there is a Z form which is in the left-handed helix orientation. Please give me some time. And then there is right-handed helix orientation and left-handed helix orientation. So there is A form and B form which is in the right-handed helix orientation, and then there is Z form which is in the left-handed helix orientation. So this DNA replication is in the semi-conservative mode, where semi-conservative mode of replication is the mode of replication of the DNA molecule. So one of the strand is continuous and one of the strand is discontinuous. So the strand which is discontinuous is known as Okazaki. The discontinuous strand is replication is in the semi-conservative mode, where semi-conservative mode of replication is the mode of replication of the DNA molecule. So one of the strand is continuous and one of the strand is hello, 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 hello. So I hope now the problem is resolved. So this is the semi-conservative mode of replication, in which one strand is continuous and other strand is discontinuous. So the discontinuous strand is known as the is consisting of the discontinuous strand consists of the lagging strand. One strand is continuous and other strand is discontinuous. So the discontinuous strand is known as the is consisting of the discontinuous strand consists of the lagging strand. So the gaps are between the lagging strand, and then there is a continuous strand which is the leading strand. So this one is the leading strand which is in the so the gaps are between the lagging strand, and then there is a continuous strand which is the leading strand. Hello. 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 Hello everyone. There was a small technical issue and I think that now it is resolved. So if you are able to hear me, then please type it in the comment box. And I think that now it is resolved. So if you are able to hear me, then please type it in the comment box. So I hope uh, now the problem has been resolved. So the DNA replication, it takes place in the 5 prime to 3 prime. It takes with the help of 5 prime to 3 prime polymerase. It reads the bases in the opposite strand and it select proper bases to put on the new strand. So it joins the new bases to the new strand with phosphodiester bond, and there is 3 prime to 5 prime exonuclease activity. There is 3 prime to 5 prime exonuclease activity, that is also known as the proofreading activity, and it reads the base pair just created for correctness. So it removes if there is some mismatching base pair if it takes place. And DNA replication begins at a specific sequence called as origin of replication. So the proteins binding at the origin, they help to open the double helix to allow the access of the replication protein. So replication, it requires the reader of a template and catalytic function to make phosphodiester bonds. So this is how DNA replication proceeds. So there is synthesis, then there is there is primer removal, then there is synthesis, and then there is proofreading. So this is a function of DNA polymerase 1. So this is a function of DNA polymerase 1. So DNA polymerase 1, it has thumb domain, it has palm domain, and it has finger domain. So there are 3 domains. One is thumb domain, one is palm domain, and another one is the finger domain. So DNA replication proteins, it consists of DNA polymerase, primase, single stranded binding protein, then there are helicases, then there is topoisomerase, then there is primer removal, and then there is DNA ligase. So this is the replication fork. So this one is the leading strand, this one is the leading strand, this one is the lagging strand. So DNA polymerase 1 is there, so it helps in the synthesis of the lagging strand. So right in the beginning the primers are added, and these are the single stranded binding protein that provides stability. This is the RNA primer that leads to the synthesis of RNA, that leads to the synthesis of the primer with the help of the primase enzyme. This one is the helicase, it unwinds the DNA, and this one is the topoisomerase, it induces nicks in the DNA. This one is the sliding clamp. So these are the different parts of the replication fork. So there is some basic difference between the prokaryotic and the eukaryotic DNA replication. So prokaryotic DNA replication, it occurs inside the cytoplasm, whereas eukaryotic DNA replication, it occurs inside the nucleus. Prokaryotic DNA replication, it only has one origin of replication per molecule of DNA, and eukaryotic DNA replication has many origins of replication in each chromosome. So origin of replication is about 100 to 200 or more nucleotides in length. So each origin of replication is formed of about 150 nucleotides, and replication in case of prokaryote, it occurs at one point in each chromosome, and replication occurs at several points simultaneously in each chromosome in case of eukaryotic DNA replication. Prokaryotic DNA replication have only one origin of replication, whereas eukaryotic DNA replication has multiple origin of replication. So in case of prokaryotic DNA replication, the initiation is carried out by protein DNA A and DNA B, whereas in case of eukaryotic DNA replication, the initiation is carried out by origin recognition complex. So topoisomerase is needed in both the cases, and replication is very rapid in case of prokaryotic DNA replication, and replication is slow in case of eukaryotic DNA replication. Then there are some sequences which are present at the end of the chromosomes, and these are known as the telomeres. So these are the chromosomes with the telomeres. So telomerase, it adds the RNA at the end of the DNA molecule which are synthesized. It binds to the 3' flanking end of the telomere that is complementary to the telomerase RNA, and so DNA replication, as we saw, it is a semi-conservative. So each one of the parent DNA strand is passed to the daughter DNA strand, and one strand for each is there. So one is the new strand, other one is the old strand. So where does and when the DNA replication takes place? So it takes place in the S phase, in the interphase of the cell cycle. So nucleus of the eukaryotes is the location where it takes place. So DNA topoisomerase is also known as DNA gyrase. So it unwinds the double helix. This is a function of DNA topoisomerase. It binds and it nicks at the 5' side. It leads to the rotation of the free 3' end. So ligase comes and this enzyme is ligated. So DNA helicase is there. This enzyme is needed for the unwinding the chain and breaking the hydrogen bond between the complementary base pairs. RNA primer adds the nucleotides for the starting point of the DNA replication. So short strands of RNA are added. As we have studied, the DNA replication is initiated at many points in eukaryotic chromosome, and they are known as the replication bubbles. So they will eventually all meet to form a whole replicated strand. So DNA duplicates in both the directions. So this is one of the direction in which DNA discontinuous synthesis is taking place. This is the strand in which the continuous strand synthesis is taking place, and these two strands are occurring in the opposite direction. This one is occurring in this direction, and it always occurs in the 5' to 3' direction. So a replication bubble is showing the old DNA strands in blue and newly synthesized strands in red. So the new strand is made only in the 5' to 3' direction. So what are origins of replication? So these are the sites along the DNA molecule where the enzyme starts the DNA replication and then proceeds in both the direction to form bubbles. This is the replication fork. This is the Y-shaped region of replicating DNA molecule where the new strands are growing. And the function of then single-stranded binding proteins come. So their function is it stabilizes the DNA strand as they are being replicated, and it prevents the rejoining of DNA strands. So single-stranded binding proteins are there. So DNA polymerase is like occurring in DNA polymerase 1, DNA polymerase 2, DNA polymerase 3. But DNA polymerase 1, it cuts off the RNA primer and fills in with DNA between the lagging strands, and DNA polymerase 3, it elongates the strand by adding DNA nucleotides on the leading strand. It also proofreads and corrects the DNA strand. So synthesis is always occurring in the anti-parallel manner as the strand builds in the opposite direction. So then there is leading strand and lagging strand of the DNA. So leading strand is the template strand of the DNA and lagging strand is the other DNA strand. So in the leading strand there is continuous addition of nitrogenous bases, and in the lagging strand there are short strands of Okazaki fragments that are synthesized. So leading strand DNA is, it takes place in the 5' to the 3' direction, and the lagging strand also synthesizes in the 5' to the 3' direction. Then there is an enzyme known as DNA ligase. So it is a linking enzyme and it joins the strand. For example, joining two Okazaki fragments together So DNA ligase is the enzyme. So DNA ligase is the enzyme that joins the Okazaki fragments with covalent bonds. So what all we studied till now is like the helicase, they unwind the parental DNA duplex. Then the single stranded binding proteins, they stabilize the unbound parental DNA. Then the leading strand is synthesized continuously in the 5' to 3' direction by DNA polymerase. Then the lagging strand is synthesized discontinuously. So primase synthesizes a short RNA primer which is extended by DNA polymerase to form the Okazaki fragment. So after the RNA primer is replaced by the DNA fragment, DNA ligase joins the Okazaki fragment to the growing strand. So this is the DNA ligase, and this one is the overall direction of replication. So there are basically 5 steps. In step 1, the helicase, it unwinds the parental DNA helix. In step 2, the single stranded binding protein, it stabilizes the unbound parental DNA. In step 3, the leading strand is synthesized continuously in the 5' to the 3' direction by DNA polymerase. In step 4, the lagging strand is synthesized discontinuously, and primase synthesizes a short RNA primer which is extended by the DNA polymerase to form an Okazaki fragment. And after the RNA primer is replaced by DNA, the DNA ligase joins the Okazaki fragment to the growing strand. So what if there is a mistake? So there is typically about 1 in a billion nucleotides which is incorrectly base paired. So proofreading function is done by DNA polymerase 1. When the initial base pairing occurs, they are usually corrected by the DNA polymerase 1. So initial base pairing errors which are occurring, so they are usually corrected by the DNA polymerase 1. Then there are telomeres which are present at the ends of the chromosome in so at the end of each chromosome is a protective cap that is known as the telomere. So each time a cell divides, the telomeres are snipped shorter. So telomerase is the enzyme which adds DNA sequence repeats. So it is TTAGGG in all vertebrates. It adds through the 3' end of the DNA strand. That is an overhang. So this is a question that what will be the complementary strand for the following DNA sequence. I am giving you time of 1 minute. You can type in the comment box. So what will be the complementary DNA strand for the following DNA sequence? So the answer is this one is the complementary DNA strand. G pairs with C and T pairs with A. So this is the ideal pairing rule that is there. So in this diagram of the process of DNA replication at the replication fork, the strand label B is the leading strand, lagging strand, template strand, Okazaki fragment or the RNA primer. So it is the lagging strand. So lagging strand is the newly synthesized DNA where the addition of nucleotides is on the ends opposite or away from the replication fork. So how we came to know that DNA is the genetic material? So there was Frederick Griffith. So Frederick Griffith in 1928, so he studied Streptococcus pneumoniae, which is a pathogenic bacterium causing pneumonia. So there are two strains of Streptococcus. One is the S strain, that is the virulent strain. Other one is the R strain, that is the non-virulent strain. So Griffith, he infected the mice with these strains, hoping to understand the difference between the strains. So what were the Griffith results? So in the live S strains, the cells killed the mice. In the live S strain, the cells killed the mice. And in the live R cell, the cells did not kill the mice. In the heat killed S strain, again it did not kill the mice. If the heat killed S strain was mixed with the live R strain cell, it killed the mice. So what was the conclusion? So Griffith's conclusion was that there is some information that is specifying virulence that which is passed from the dead S strain into the R strain. So this is where the idea came that DNA can be the genetic material. So Griffith called this transfer of information as transformation. So the virulence that is passed from the dead S strain into the live R strain, then Avery, MacLeod and McCarthy in 1944, they repeated Griffith experiments using purified cell extracts, and they discovered that removal of all proteins from the transforming material did not destroy its ability to transform the R strain cells. So Avery, MacLeod and McCarthy, they repeated the Griffith experiment using the purified cell extract, and they discovered that removal of all proteins from the transforming material did not destroy its ability to transform the R strain cells. So DNA digesting enzymes, they destroyed all the transforming ability, and the transforming material is the DNA. The transforming material is the DNA. Then Hershey and Chase came in 1942 and they investigated bacteriophages. So bacteriophages are the viruses that infect the bacteria. So bacteriophage was composed of only DNA and protein, and they wanted to determine that which of these molecules is the genetic material that is injected into the bacteria. So they wanted to identify that which one is the genetic material. Is the protein the genetic material, or the DNA is the genetic material that is injected into the bacteria? So in this case, the bacteriophage DNA was labeled with radioactive phosphorus, that is the P32, and the bacteriophage protein was labeled with radioactive sulfur, that is S35. So radioactive molecules were tracked. So only the bacteriophage DNA entered the bacteria, which was used to produce more bacteriophage. So only the bacteriophage DNA, as indicated by the P32, it entered the bacteria and it was used to reproduce more bacteriophage. So it clearly shows that DNA is the genetic material. So the conclusion was that DNA is the genetic material. So this was the case where the conclusion came that DNA is the genetic material. So something about the structure of the DNA molecule. So DNA is a nucleic acid. So building blocks of DNA are the nucleotides, and each of them are composed of a 5' sugar, a 5 carbon sugar called as deoxyribose. Then there is a phosphate group, and there is a nitrogenous base. So nitrogenous base, phosphate group, and 5' sugar molecule called as deoxyribose, they all form the nucleotides. So they are the building blocks of the DNA. So adenine, thymine, guanine, and cytosine are the nitrogenous bases. So adenine and guanine are the purines, whereas cytosine, thymine, and uracil are the pyrimidines. So nitrogenous bases are of two types. One is purines, and another one is pyrimidines. And the 5 carbon sugar is also of two types. One is deoxyribose, and other one is ribose. So in the deoxyribose, one oxygen is deleted from here, which is not deleted in case of ribose. So ribose occurs only in the RNA, and deoxyribose, it occurs only in the DNA. So nucleotide structure, it consists of the nitrogenous bases that are attached to the 1' carbon of the deoxyribose. Phosphate group is attached to the 5' carbon of the deoxyribose, and the free hydroxyl OH is at the 3' carbon of deoxyribose. So there is a nitrogenous base which is attached to the 1' carbon of the deoxyribose sugar, and the phosphate which is attached to the 5' end of the deoxyribose, and then there is the free 3' OH group, 3' hydroxyl group, which is free in nature. So this is the structure of the nucleotide. So nucleotides are connected to each other to form long chain. So phosphodiester bond is formed between adjacent nucleotides, and it is formed between the phosphate group of one nucleotide and the 3' OH of the next nucleotide. So phosphodiester bond is formed between adjacent nucleotides. It is formed between the phosphate group of one nucleotide and the 3' OH of the next nucleotide. So the chain of nucleotide has a 5' to 3' orientation. So this is the phosphodiester bond. Which is formed between the 3' and the 5' of the deoxyribose sugar. So in determining the three-dimensional structure of the DNA, it involved the work of few scientists. So Erwin Chargaff determined that the amount of adenine is equal to the amount of thymine. So Erwin Chargaff he determined that the amount of adenine is equal to the amount of thymine, and amount of cytosine is equal to the amount of guanine. So this is known as the Chargaff rule. Then Rosalind Franklin and Morris Wilkins, they performed the X-ray diffraction studies to identify the three-dimensional structure. and they discovered that the DNA is helical. They discovered that the molecule has a diameter of 2 nanometer and makes a complete turn of the helix every 3.4 nanometers. And in 1953, James Watson and Francis Crick, they deduced that the structure of DNA using the evidence from the Chargraph, Franklin and others proposed a double helix structure. So they proposed a double helix structure. So double helix consists of two sugar phosphate backbones. It consists of a nitrogenous base that is towards the interior of the molecule, and it consists of bases which form hydrogen bonds with complementary bases on the opposite sugar phosphate backbone. So there is this 5' to the 3' end, and then this is the anti-parallel strand from the 3' end to the 5' end. This one is the sugar phosphate backbone. So the two strands of nucleotides, they are anti-parallel to each other. So one is oriented in 5' to 3' direction, and the other is from the 3' to the 5' direction. So the two strands, they wrap around each other to create the helical shape of the molecule. So the two strands wrap around each other to create the helical shape of the molecule. Then came Messenschel and Stahl in 1958. They investigated the process of DNA replication and considered three possible mechanisms. One is the conservative model, other one is the semi-conservative model, and third one is the dispersive model. So DNA replication is semi-conservative and discontinuous mode of replication. So to deduce this, so the bacterial cells were grown in the heavy isotope of nitrogen, that is N15. So they all incorporated the N15 DNA. Then cells were switched to a medium containing lighter N14 N14 isotope. So DNA was extracted from from cells at various time intervals. So DNA from different time points were analyzed for the ratio of N15 to N14 it contained. So after one round of DNA replication, the DNA consisted of N14-15 hybrid DNA molecule. And after two rounds of replication, the DNA contained two types of molecule. One is half the DNA was N14 N15 hybrid, and half the DNA was composed of N14. So Messenschel and Stahl, they concluded that the mechanism of DNA replication is semi-conservative model. So each strand of the DNA, it acts as a template for the synthesis of a new strand. And DNA replication, it includes initiation, where replication begins at the origin of replication. Then there is elongation, where new strands of DNA are synthesized by DNA polymerase. And then there is termination, where replication is terminated differently in prokaryotes and eukaryotes. So chromosome of a prokaryote is circular molecule of DNA, and replication begins at one origin of replication and proceeds in both the direction around the chromosome. So this one is the origin of replication, and this one is the origin of termination. So replication occurs in a bidirectional manner, and two replications of origin can be seen. And finally, two daughter DNA molecules are synthesized from one parent DNA molecule. So prokaryotic DNA replication, the double helix is unwound by the enzyme helicase. DNA polymerase 3 is the main polymerase that is responsible for majority of DNA synthesis. DNA polymerase 3, it adds the nucleotide to the 3' end of the daughter strand of the DNA. So this is DNA polymerase 3 in action, where it adds the nucleotides at the 3' end of the DNA molecule. So new strand is synthesized from the 5' to the 3' direction, and this one is a sugar phosphate backbone. Template strand is from the 3' to the 5' direction, whereas the new strand is grown from the 5' to the 3' direction. So both the strands are anti-parallel. So DNA replication is semi-discontinuous. DNA replication is semi-discontinuous, where polymerase 3 can add nucleotides at the 3' end of the newly synthesized strand. And DNA strands are anti-parallel to each other. So leading strand is synthesized continuously in the same direction as that of the replication fork, and lagging strand is synthesized discontinuously, creating Okazaki fragments. So if you have any doubt, any queries, I'm sorry for the technical glitches that happened today in today's session. So I will try to correct it and make sure that it doesn't occur again. And DNA strands are anti-parallel to each other. तो अगर आप लोग के पास कोई क्वेरी है तो यू कैन टाइप इट इन द कमेंट बॉक्स. Yes, Samajit Patra, what we will read for the next class and when will be the next class? So next class will be tomorrow again, 6 p.m. will be the time when the next class will be there. Good evening everyone. Good evening to Nitesh Kumar Gupta as well. So good evening to all of you. So आप लोग DNA replication पढ़ सकते हैं and you can study DNA replication for the next class because I will be asking the questions from there only. So Samajit Patra is asking how many minimum units we have to prepare for cracking CSIR UGC NET sir. So अभी का क्या हो गया है? If you see this year's paper and अब पेपर एक साल पहले का भी पेपर आप देखोगे, so अभी सारे मॉड्यूल्स को पढ़ना इम्पॉर्टेंट है बिकॉज क्वेश्चंस आर आस्क्ड इन द इंटरलिंक्ड मैनर. सो आप पहले की तरह अब ऐसा नहीं कर सकते कि यू कैन जस्ट स्टडी फ्यू मॉड्यूल्स एंड लीव द रेस्ट मॉड्यूल्स. सो आपको लगभग सभी मॉड्यूल्स को आपको अच्छे से करना होगा. दिस इज अ गुड क्वेश्चन दैट हाउ मेनी क्वेश्चंस वी अटेम्प्ट मैक्सिमम. सो in section C you have to attempt 25 questions, so try to attempt something around 20 to 22 questions minimum absolutely correctly in the section C. So मैं एक separate video बनाऊंगा strategy पर that how many questions you have to attempt and what all you should do, and then in section B there are 35 questions which you have to do which are carrying 2 marks each, and then there is section A where 15 questions need to be done. So that is again of 2 marks. So the ideal strategy should be to attempt maximum questions from section C. आप लोग ज़्यादा से ज़्यादा questions करिए from section C and then followed by section B and then followed by section A. If you are good in section A, if you are good in aptitude and reasoning ability, then you can go for section A as much as maximum questions, like out of 15 you can go for 12 or 13 questions. And in section B you can go for something around I would suggest that you, if you are not that confident in attempting the section B questions, then you should go somewhere around 20 to 25 questions. So this much of questions are enough if you do it correctly to crack the CSIR. So आप ये भी कर सकते हैं कि आप हम लोग के साथ जुड़े रहिए और हम लोग की क्लासेस देखते रहिए. मैं और कुमकुम मैम क्लासेस लेते हैं लाइफ साइंस की. सो और हम लोग मॉर्निंग में MCQ सेशन कराते हैं जो कि 11:30 AM पे होता है. वो हमारा MCQ सेशन होता है. एंड शाम को हमारी थ्योरी वाली क्लासेस होती हैं जो कि अभी चल रही हैं. दैट इज़ ऑन द 6 PM क्लास. सो दैट इज़ ऑल आई कैन टेल यू राइट नाउ. ओके समजीत. हाउ टू डाउनलोड क्वेश्चंस ऑफ प्रीवियस ईयर. आप लोग NTA की वेबसाइट पे जाइए और उसमें आप लोग you can go and download the question paper from there. So I will make a separate video that how to download the question paper and how many questions one should attempt. So इसके लिए strategy session separate session होगा. So I will take a separate session on that one. So you can download by going to the NTA website. बुशरा खान ने पूछा है सर प्लीज़ टेल अबाउट द टेस्ट सीरीज फॉर जून 2021। सो बुशरा आप हमारे साथ बने रहिए, हम लोग बहुत ही जल्द टेस्ट सीरीज, मैं अपने कोर्स में टेस्ट सीरीज इंक्लूड करूँगा जो कि मैं प्लस पे लूँगा, और मैं वैसे भी इम्पोर्टेंट क्वेश्चंस डेली 11:30 AM पे करा रहा हूँ। सो आप हमारी वाली क्लासेस अटेंड करते रहिए, उससे आपको बहुत फ़ायदा होगा। और इफ़ यू वांट टू डू टेस्ट सीरीज सेपरेटली, then one thing is important for the test series, you can solve as many as questions, but अगर आपका concept सही है then with the minimum of effort you can crack this examination. So right now your focus should be to complete the syllabus. अभी आपका focus होना चाहिए बुशरा to complete the syllabus, और syllabus को अच्छे से complete करने के बाद then you should focus on solving the questions. हम लोग के साथ आप बने रहिए, हम लोग यहाँ पर बहुत सारे questions लेकर आए हैं। मेरा सेशन होता है 11:30 AM पे, जहाँ पे मैं क्वेश्चंस डेली सॉल्व कराता हूँ। सो आप वाला सेशन अटेंड करिए, सो वहाँ से आपको काफ़ी आइडिया लगेगा, एंड मैं इंपॉर्टेंट क्वेश्चंस ही कराता हूँ। सो दैट इज़ द थिंग। सो एंड अबाउट द टेस्ट सीरीज, सो टेस्ट सीरीज आप एग्ज़ाम से 1 month एंड 1.5 months पहले आप स्टार्ट करिए। आप चाहें तो रेगुलरली भी टेस्ट सीरीज कर सकते हैं। इफ़ यू go for my plus course, then you will be getting unit wide test series which you can attempt regularly. So right now we were doing DNA replication. So the enzymes of DNA replication, सम्मिजित पात्रा ने पूछा है कि सर कैन यू प्लीज़ टेल मी अबाउट यूनिट वाइज बुक्स फॉर CSIR UGC नेट। सम्मिजित, मैं एक सेपरेट स्ट्रैटेजी वीडियो लेकर आऊँगा जिसमें मैं सारी चीज़ें डिस्कस करूँगा, that what are the sources from where one should study, और वो बहुत ही जल्द होगा वो स्ट्रैटेजी वीडियो। सो आप उस स्ट्रैटेजी वीडियो को देखिएगा जहाँ से आपको पता चलेगा। मैं बहुत ही जल्द वीडियो बनाऊंगा उसके ऊपर, जो कि आप देख पाएंगे यूट्यूब पर। सो अगर अभी के लिए आपको पढ़ने के लिए चाहिए तो जैसे बायोकेमिस्ट्री का यूनिट है तो यू कैन गो फॉर लेनिंजर और वॉइट एंड वॉइड। इफ़ यू वांट टू गो फॉर cell biology, then you can go for Gerald Karp, or you can go for there is one book by molecular biology of the gene, and you can go for Cooper. Then there is one book by Cooper. Then for unit 3 you can go for Benjamin, Pierce and Watson. For unit 4 you can go for Lordish or Cube. For unit 5 you can go for Gilbert. For unit 6 you can go for Tegan Zeiger. For unit 7, which is animal physiology, you can go for Vander's physiology. For unit 8 you can go for Benjamin, Pierce. For unit 9 you can go for NCRT. For unit 10 you can go for Peter Stilling. 11 के लिए Streckberger, 12 के लिए TA Brown, एंड 13 के लिए Primrose and Wilson and Walker, एंड पार्ट ए के लिए प्रीवियस ईयर क्वेश्चन। सो हम अपनी आने वाली सीरीज़ में सारे इंपॉर्टेंट क्वेश्चंस और एक स्ट्रैटेजी सेशन के साथ हम आपको बताएंगे कि विच बुक शुड यू रीड। ओके, सो आई विल क्रिएट अ न्यू वीडियो। So thank you everyone for today's class, and I'll meet you tomorrow. कल हमारी क्लासेस फिर से होंगी 6 PM पे हमारी क्लासेस होती हैं, और 6 PM पे मंडे से फ्राइडे हमारी डेली क्लासेस होती हैं। एंड देन हमारी क्लासेस आप अटेंड कर सकते हैं 11:30 in the AM in the morning, जहाँ पे हम MCQ प्रैक्टिस करेंगे। सो आप लोग कल के लिए यू कैन गो ऑन एंड यू कैन रीड द DNA replication in detail, and if you have any doubts or if you have any queries then you can post in the comment box, and I will answer them. So thank you. I am closing the session.